Hello everyone, my name is Dr. Farah Maarfi and you are welcome to the class of biotechnology. Today we will discuss about the nuclease, specifically to the restriction enzyme that act as a molecular scissor. But before we move forward, please like and subscribe to this channel so that you cannot miss any of the future video. Nucleases are of two types, endonucleases and exonucleases. They both can cut down or break down the nucleic acid. Nucleic acid is DNA or RNA. So it can cut down the DNA or RNA by cleaving its phosphodiester bond between the nucleotides. How it's cleaving out or cutting down the DNA or RNA molecule? By breaking down its phosphodiester bond. Suppose this is a DNA molecule and when you treat it with some nuclease, it will cut down the, or you can say, chop down the DNA molecules into different fragments. Now you get here two fragment 1, fragment 2, right? It's a one single DNA, and after treating it with nucleases, you get a two fragments of a single DNA. So how it is cutting it into two fragments? By chopping it down or by targeting its phosphodiester bond. Where it is present? It is present in between the two nucleotides. For example, suppose this is a chain of a DNA molecule A, T, G, C, T, A, 5 prime, and from this side it would be T, A, C, G, A, T, 3 prime. So this position, at this position, this is the phosphodiester bond. It is present between the two nucleotides. Here it is present between thymine and guanine. When you treat it with nucleases, suppose nucleases cut it at somewhere in between this point. So after treatment, you will get the fragment like this: 3 prime, 5 prime, T, A, 3 prime. And from this side, it would be like G, C, T, A, 5 prime, 5 prime, C, G, A, T, 3 prime. This whole process is called hydrolysis. During this process, nucleases utilizes the water molecule to cut down the DNA into fragments. Nucleases can create single or double stranded breaks in their target molecule, and they are of actually the type of enzyme that called the hydrolase enzyme. The nucleases are actually a hydrolase enzyme. These are hydrolase enzyme. Since you are now aware that nucleases are of two types, that is exonucleases and endonucleases, let's understand how they are being different from each other. So if we talk about the exonucleases, exo means outside or at the ends. So it catalyzes the hydrolysis of terminal nucleotides. Suppose this is a DNA molecule. This starts from the 3 prime ends, and this one starts from the 5 prime end and ends at 3 prime end, and this one ends at 5 prime end, right? So if we treat this particular DNA molecule with exonuclease enzyme, this is an exonuclease enzyme, and if we treat this DNA molecule using this enzyme, then it will cleaves the DNA molecule at the end of these strands, either starting from the 3 prime to 5 prime ends direction, or from the 5 prime to 3 prime direction. As it is said here, exonucleases catalyzes hydrolysis of terminal nucleotides, right? It catalyzes the hydrolysis of the terminal nucleotides. That means it cuts down or it cleaves out the molecules or the nucleotides at the end of the DNA strands or RNA strand, right? either 5 prime to 3 prime direction or 3 prime to 5 prime direction. Exonuclease first and exonuclease second are the example of exonuclease enzyme. If we talk about endonucleases, it is a site-specific endonucleases that it can recognize a specific base sequence. That means it is restricted to some specific base within the DNA or RNA molecule and makes a cut internally, not at the ends or terminals, but it make a cut internally. So it recognizes the DNA molecule in the internal backbone of the DNA or RNA molecule and makes a cut over there where internal phosphodiester bond, where at internal phosphodiester bond within DNA molecule. Eco R1, BAM H1, Hind third, these are the examples of the endonucleases. Suppose this is a DNA strand and this is a endonucleases restriction enzyme, right? It is a DNA molecule and this one is the endonuclease restriction enzyme. And if we treat this DNA molecule with the endonucleases or restriction enzyme, then it first recognizes the particular strand or particular bases where it should make a cut. So first it recognizes its own site, own particular site, then make a cut. Restriction endonuclease enzymes are encoded by bacteria and archaea. That means you can find these in bacteria and archaebacteria, right? So there it provide actually a defense mechanism against the viruses. Suppose this is a bacterial cell, it's a genetic material, and this one is the plasmid present inside the bacterial cell, right? This is bacterial cell. Bacterial cell. Suppose some virus, for example, bacteriophage, you can say, comes to attack on it, right? Then the restriction enzyme present inside this bacteria provide protection against this virus by chopping down its genetic material. So as soon as this particular virus get adhered on its surface like this, it tries to transport its genetic material, that is RNA inside the bacterial cell. But as soon as this restriction enzyme recognizes the presence of some foreign material, that is the RNA of the virus, it recognizes it and it chopped it down or it cuts it down into fragments and hence stop the translation process. By this way, it can provide a defense mechanism to the bacterial host against the invading virus. Invading virus, that is the bacteriophage. Now a question arises, if restriction enzyme present in bacteria and it is the property of the restriction enzyme to cut down the DNA or RNA into fragments, then why it is not cutting its own DNA, right? Because inside the bacteria, restriction enzyme is also present there and the genetic material DNA or RNA is also present there. But why it is not cutting down its own DNA molecule? So the answer is that in the bacteria, restriction enzymes form combined system. It forms combined system. Combined system of what? Restriction modification system. Restriction modification system. It combines with the modification enzyme, that is methyl transferase. There it methylates the bacterial DNA. So as soon as the bacterial DNA gets methylated, then if it is recognized by the restriction enzyme, So due to the presence of this methyl group on the DNA molecule or the RNA molecule, restriction enzyme thinks it is its own DNA. So it inhibits its cleavage. It does not cleave the or it does not proceed the process of hydrolysis. In the year 1970, the first restriction enzyme, that is HIN second, was isolated. It is the first restriction enzyme. First restriction enzyme. It is isolated in the year 1970, and after that, in the year 1978, Daniel Nathans, Warner Arbour, and Hamilton O. Smith, these three scientists were awarded by Nobel Prize in Physiology or Medicine. Since then, these restriction enzymes are having importance or significant role in the recombinant DNA technology. The restriction endonucleases are named according to the organism in which they were discovered. So they are named according to the organisms in which they were discovered, right? By the system called a letter number system. Letter and number system. This is a system using which we can name the particular restriction enzyme in the organism as per the organism in which it is discovered. For example, third, right? So here H represents the Haemophilus, that is the genus. I and N represents the species. D here represents the strain, and third here represents the order in which it is being isolated from the particular organism. It is the order. Let's see few other examples like Eco R1. In Eco R1, E is the genus, that is Escherichia, and CO represents the coli. R here represents the strain, and I is the order of its isolation, right? In which order it is being isolated from the particular organism. In HIN third, as we already discussed, H here represents the Haemophilus, IN represents the influenza, D here represents the strain. Influenza is the species, Haemophilus is the genus, and third here represents the order of its isolation. For BAM-H1, B represents the Bacillus, AM represents the Amylo for the species, H here represents its strain, and I here represents the order of its isolation. There are several other examples of it also. Now moving forward to understand the working mechanism of the restriction enzyme. As the matter of fact, you all know that once the restriction enzyme recognizes the restriction site or the specific basis, it makes a cut over there. So it makes a cut in two different ways. One by producing the sticky end and another is by producing the blunt end. Suppose this is the sequence of DNA molecule, right? Eco R1 is an example of restriction enzyme that produces the sticky ends only, right? So for the restriction, for the Eco R1, EAATTC, this is the specific recognition site. It is the specific recognition site for Eco R1 only. Eco R1 site. This is the Eco R1 site where the Eco R1 enzymes comes and attached to it. So it makes a cut in between the G and A. It recognizes, Eco R1 recognizes this site. Eco R1 recognizes this GAATTC and makes a cut in between the G and A at phosphodiester bond. In this strand here and in this strand at this point. This is a palindromic sequence. This type of sequence is called the palindromic sequence. It is an mirror image of each other. Eco R1 is an example of it. So after making a cut, fragments like this, and this way it produces a sticky ends. Sticky ends means it generates flanking regions or hanging regions in both the strand of a single DNA molecule, right? This is the example of sticky end. Another example is BAM-H1 and HIN3rd. So many other examples also. BAM-H1, HIN3rd. They also produces the sticky end in the same manner. Talk about blunt end. Then the examples are ISMA first, ECO R1, 5. It is ECO R1, 5. ISMA 1. So depending upon the type of the restriction enzyme, you will get the sticky end or blunt end. So ISMA first, ECO R1, 5. These produces the blunt end. Suppose this is again this is the strand of the DNA molecule, and this is the site for ECO R1, 5. GAT ATC. So when you make a cut off using ECO R5, then enzyme comes. For example, this is an enzyme. In it comes, recognizes GAT ATC, make an attachment over there, recognizes the phosphodiester bond that is present between A and T. Here. So it makes a cut in between the A and T like this. It separates the molecule into two different fragments of same length. Will not get any hanging or flanking regions. You will get the blunt ends only when you treat with ECO R5. So after treatment with ECO R5, you will get the blunt end. The table here represents the name of the different restriction enzymes from the source it is obtained, then the recognition site by which it recognizes the specific basis where it makes a cut, and then the nature of the cut, that whether it would be sticky end or the blunt end. Based on the enzyme subunit composition, cofactors, nature of the targeted sequence, and the position of the restriction site, restriction enzymes are of 4 types: type 1, type 2, type 3, and type 4. Let us understand each of them one by one, starting with the restriction enzyme. First, restriction enzyme first consists of 3 proteins that function as a single protein complex. So this is a 3 protein complex actually. It is a protein complex that functions as a single unit. The complex specifically consists of 3 different types of protein, that is specificity protein. Specificity protein. This pink color subunit represents 2 subunits of methyl transferase. Methyl transferase. One is specificity protein, another is methyl transferase, and third is 2 restriction endonuclease subunit. Restriction endonuclease subunit. So these 3 proteins all together forms a protein complex and it acts as a single unit. It acts as a single unit. As this restriction enzyme recognizes the DNA sequence, it cuts at a site far from the recognition site, and during the recognition and the cutting or the cleavage process, it requires ATP and Mg2 plus as a cofactor and ATP for the energy. It also required SAM, S adenosyl methionine. Once the protein complex binds the DNA molecule, it acts as a molecular motor. Utilizing the ATP, it translocates thousands of base pair. It translocates thousands of base pair until it encounters another complex. And at this point, it creates a double-stranded break. So this is the complex first, and it keeps on translocating the thousands of base pairs until and unless it encounters the second complex or the position where it binds to the DNA molecule. And as soon as it recognizes the second complex, it creates a cleavage. As cleavage occurs after translocation, the cleavage site is not fixed. As a consequence, there is no distinct cleavage pattern. So, there is no distinct cleavage pattern. Since the cleavage pattern is not distinct, we are not often using this type of recognition enzyme for the cloning purposes. So this was the working mechanism of the restriction enzyme first, that how it makes a cut and produces no distinct cleavage pattern. But unlike to this restriction enzyme second and third, they are a little bit different from the restriction enzyme first. But we will see the restriction enzyme second, third and fourth in the next portion of this video. For today we will stop here, and if you like this video, please like and subscribe and continue watching this video for the next series.